Swayam Prabha. Digital India. Educated India.
Welcome back to the fifth lecture of the ninth week. So, in the last class, I left you with that puzzle situation where I gave you this image of your lecture 5, week 9. So, I gave you that puzzle situation where I told you that you can make a figure, you can make an image by having either the background dark or the foreground dark. Okay. So, here is a case of foreground is dark and here is a case of background is dark. And I told you use this dark background concept to understand the retina. So, now in the light of this, look at this image. So, what is happening here? So, if this is the image which has to be formed, so what will happen? This place will remain, the currents will be following out here, and on these spots current will not follow. And this will create a typically a pattern like this, what is there. This is how, now coming back to the picture where I digressed to make you understand this, what you see out here is that dark current out here. Okay, so this is the dark current at minus 40 millivolt. So whenever light shines, so now let's correct it. Light shines and the membrane goes to minus 80 millivolt. And so once it goes to minus 80 millivolt, there is no current. Because at minus 80 you know that you cannot have any depolarizing pulses functioning. Okay? So now how this current flow is regulated biochemically, that is a very critical step to understand. So as I told you, I will take the example of rhodopsin or rods that holds true for the conopsins too. So coming back to the rhodopsins. Okay. Okay. If you look at rhodopsin, it is made up of a opsin protein and a retinyl moiety, which is in a normal situation it remains in a cis configuration. Okay. So those of you remember there's something called a trans and there is something called a cis. So this is your cis and the trans is like this. Okay. So now they remain in a kind of a retinyl moiety clubbed into an opsin protein. Under the dark situation, when it is generating current, so when it is generating current, it means it has these sodium channels which are present on the surface. Okay. These sodium channels remains open. Okay, they remain open. How they could remain open? Because as of now we talked about that they have a very sharp kinetics, like they open and they closes. So it is something that you imagine, whichever room you are sitting, there must be a door, right? Just think of it. You kept the door open by, how you can keep the door open? You can keep the door open by having a door stopper. Suppose it's a door which closes by itself, like it has the hinge, kind of it closes. But if you open the door and put the door stopper, then the door will remain open. Or on the side you have these door stopper, the different kind of door stoppers. You can keep the door open. So this sodium channel has a door stopper. Okay, and this door stopper of sodium channel, which keeps the sodium channel open, is cyclic GMP. So it seems now so there is a door stopper like this, cyclic GMP. So this is holding that door open in the dark. Mind it, in the dark, when the cell is maintaining a membrane potential of minus 40 millivolt. Okay, up to this it's clear. Now step 2. So listen to me very carefully. There is only a handful of informations here. Once this gets clear to you, everything will make sense. Now the next situation is when the light falls, and when the light falls, what we see, the end result we know is the membrane potential from minus 40 goes further negative to minus 80, and it prevents the flow of current. Fair enough? So now let's put this. Light is falling. As soon as the light falls, so I told you that most of the rhodopsin molecules are sitting here. In the case of cones, these are called conopsin molecules. These are light sensitive pigments which are sitting there. These rhodopsin molecules, after receiving the light, there is a change which happens. So this is rhodopsin, and there is a light falling on rhodopsin, and this split rhodopsin into opsin plus retinyl moiety, which originally was in cis, then becomes trans. Okay. Now in the meantime, what this opsin does, this opsin, adjacent to it, there are proteins called transducin. They activate the transducin. Otherwise, transducin remain in an inactive form. They activate the transducin. This transducin, on its membrane, there is another protein which is sitting there, which I am showing in green color. It activates phosphodiesterase enzyme. These activated phosphodiesterase called PDE, phosphodiesterase, then goes to these door stopper, the cyclic GMP, and dissociate it. And as soon as phosphodiesterase dissociated the cyclic GMP, the door of sodium channel closes. And once the door of sodium channel closes, the next thing you know is the membrane gets back to minus 80 millivolt. So let's recap what all I talked to you. Light falls. The rhodopsin molecules split into two parts, opsin and retinal moiety. Retinal moiety, which otherwise remain in a cis configuration, become trans configuration. Opsin, on the other hand, activates transducin, which is a very adjacent protein sitting there. Transducin activates phosphodiesterase. Phosphodiesterase goes and dissolves the gate of cyclic GMP, and sodium channel gate, which was open, which was kept open by a stoppercock or a door stopper called, not stoppercock is the wrong word, the door stopper of cyclic GMP is removed, thereby closing the sodium channel, and the membrane voltage falls down to minus 80. Now, what happens to this retinal, transretinal? There is an further isomerization. This is called the bleach cycle. It again comes back to a cis by an enzyme which transforms it from trans to cisretinal, and in the meantime it again recovers the opsin, and it again from rhodopsin. So this cycle goes on. So this is called the classic bleaching cycle, and this whole current interplay of dark and light, what we talked about, is how the image is formed. So now whenever this dark current it synapses, it sends the impulse to the next layer, which sends to the next layer, which sends it to the next layer, and likewise, and so goes on. So now when the grossly, if we look at it now again, when these rods and cones started to die off, which is the third level of damage, what we talked about in the eyes, your options are very limited. Either you have a therapy by virtue of which you can regenerate or save the dying of these cells, regenerate rod and cone photoreceptors, PR is photoreceptors, or your other option is that you completely bypass the eye. What you do, you put a camera instead of the eye and kind of, get the camera fixed up with the optic nerve. So you do not need to use the eye. So you have a camera here which will take the image and will feed directly to the optic nerves, and the optic nerves will carry the message. And if you want to look this kind of research what people have done or currently doing big time, you should visit this gentleman's website, Mark Humayun from University of Southern California at Los Angeles. So he has been working in this area. Okay, where visual prosthesis has been taken to a different height. So you can even interface the camera with the visual cortex directly, completely bypassing the optic nerves. Well, in terms of the damage to the optic nerves, where the visual signal has to travel to the visual cortex, the only possible therapy what mankind is searching for will be if some way or other, by using stem cell therapy or some way or other, we can, restore the connectivity. That is the other options. So if you remember, when we talked about the eye, there are different level of blindness we talked about, right? So we first talked about the blindness of the cornea. Again, circle it, give you a brush up in the lecture 1 of the 9th week. Next, we talked about the lens, second one, right? This is cornea, followed by lens, followed by a retina, followed by optic nerves. So I told you that in this situation, if I had to put it, so your option is that you interface these optic nerves, assuming the optic nerve is all working fine, with a camera. If you see the gross picture, that will make sense to you how you really can do that. Okay. Or, I told you, if your optic nerves going to the brain, if these optic nerves are damaged, then your option is regenerative medicine, or some way or other, some certain cells, certain of these neurons has to regenerate, or some stem cell has to replace them. Okay. So these are the different level or control point where you can interface or you can understand this whole retinal image processing. So at this point, what I will recommend is go through the whole week lecture in one go, think over it, because I kind of spent almost 5 lectures on it, because this needs a lot of understanding of a 3-dimensional geometry, how things are arranged out here. Because unless otherwise you understand once and for all, and that's especially the concept of the dark current, and how if your foreground is dark, how you can still get an image. If your foreground is active, still you can make an image. These things needs a lot of understanding, and moreover, a lot of thinking process has to go in your brain, like how this is possible. Otherwise, things will become blurred. So with this, I will close in on the nervous system, and next week we'll be starting with the cardiac system and followed by the digestive endocrine, because these things will be much more easier in the light of what all we have already covered. So please go through it, and all the best, and I am closing this lecture for the 9th week. Thank you.